Hello, my name is Tammy Wallace, and today we will be discussing the cell cycle of eukaryotic or somatic cells. These are cells that are found within any living organism, sans the sex cells. As cells grow, they move through the cell cycle. As with anything in nature, from us as humans to the seasons, there are cycles with distinct patterns and phases that occur. Cells are no different. From this figure, we can see that cells live their lives moving through their cycle. Each phase is represented by a different color and notated with a different name, or in this case, a letter. Though there are 4 total phases, the cycle is divided into 2 major parts. In the gray, the majority of the cycle is seen here, the interphase. The second section is the M phase, seen here as a pie shape with the green arrow. During interphase, or the time between divisions, many of the things are accomplished, such as photosynthesis, cell respiration, protein synthesis, and DNA synthesis. The actual division portion of the cell occurs during the M phase, where mitosis and cytokinesis occurs. A family of protein kinases controls the timing of the cell cycles that act due to changes in response to cellular signals. The metabolic activities within the cell cycle are done via phosphorylizing specific proteins, allowing for precise intervals and activities within each of these cell cycle phases. The protein kinases are cyclin-dependent protein kinases, or I'll be referring to them as CDK, with a regular cyclin subunit. Eukaryotic cells go through a series of phases during active division, thus the cell cycle. The last slide shows that each stage is labeled with varying letters. Each letter represents a different phase. The G phase represents the 2 gap phases, G1 and G2, shown here and here. And the S phase is the synthesis phase, where genetic material is being duplicated. And lastly, the M phase, notating the mitosis phase. As previously stated, interphase is the section of cell division that encompasses both gap phases and synthesis phase. Starting interphase is G1, or the first growth or gap phase. Note that this is the longest of the cell cycle, roughly taking 6 to 12 hours to occur. Here, the cell grows and nearly doubles in size. During this phase, the cell has 2 options, either division or cease to divide by entering the GO or G0 resting state. This is seen here. GO cells, such as specialized cells like muscle or nerve cells, no longer need to divide, and therefore they remain in the G0 state or phase to perform their specific functions. Thus, these cells are considered to be terminally differentiated and will remain in that G0 state. However, when other cells are ready to divide again, they re-enter the G1 phase. G1 is the state responsible for RNA and protein synthesis, allowing the cell to grow while performing its specialized tasks. Within the G1 phase, there's a point known as the restricted point or checkpoint, located roughly here. This is the pass of no return, or the point of no return, so to speak. Here the cell is committed to moving to the next phase of the cell cycle, such as the S phase. The S phase, or synthesis phase, the second largest phase of the cell cycle, lasting roughly 6 to 8 hours, is where the DNA synthesis occurs. Here the amount of DNA is roughly doubled, and as synthesized, the RNA and proteins continues. The genetic material duplicates, allowing each chromosome to have its new sister chromatid form. Once all of the chromosomes are duplicated, the cell moves into the G2, or second gap phase. DNA synthesis ceases within the second gap phase, while RNA and protein synthesis still continues. This phase takes roughly 3 to 4 hours. Metabolic changes allow for required cytoplasmic material for mitosis and cytokinesis to form. G2 also is responsible for ensuring that all of the duplicated chromosomes are error-free, making necessary corrections before entering into M or mitosis phase. This is where the G2/M checkpoint comes into play. Again, located roughly right here. Again, this ensures that the cell is ready to move on to the next phase while maintaining genomic stability. Once the cell has completed its growth phase or interphase, here, which fun fact is roughly 90% of the cell cycle, the cell is ready to start the division cycle or phase, being mitosis. Mitosis is the division portion of the cell cycle that produces two daughter cells containing the same genetic information as the original parent cell. Mitosis is divided into phases, the first of which is prophase. Prophase is a large portion of mitosis and can be broken down into phases. As you see here, we have prophase and then again late prophase. Prophase breaks down the nuclear membrane of the cell to form small vesicles. At this time, the nucleolus disintegrates. The centrosome duplicates to form two daughter chromosomes, centrosomes, and they start to migrate to opposite ends or poles of the cell. Spindle fibers are produced to constitute the meiotic spindle, and the chromosomes condense into compact structures. The replicated chromosomes consist of two identical chromatids that are held together at the centromere. The centrosomes and thus the chromosomes migrate to the cell's equator, creating what looks like right angles to the axes. At the kinocore, the spindle fibers bind with centromere and each chromosome and chromosomes continue to condense. The next phase being mitosis. The chromosomes align themselves along the equator, which is also referred to as the metaphase plate at the spindle apparatus. The centromeres then start to divide during the anaphase portion, the next portion here, of mitosis, which allows for the sister chromatids of each chromosome to be pulled apart by the spindle fibers attached to the kinocore regions. You can see here we started with X's and now we have the V's in upside down V structures. During this phase, the cell essentially reverses then we go into telophase, and this is where most of the observed things that have occurred throughout prophase are now being reversed. The nuclear membrane reforms, the chromosomes uncoil and diffuse, and the spindle fibers disappear. Now we have what looks like two identical conjoined twin cells. This is where cytokinesis comes into play, the final portion of cellular division within the cell cycle. The cell divides into two separate cells, allowing for the cells to re-enter the interphase portion of the cell cycle. So here we have those two conjoined twins. Cytokinesis splits those two in half, thus giving us two separate daughter cells where each of them can go right back into the cell cycle. Now that we know the various stages and phases of the cell cycle, we need to learn what allows the movement from one phase to another. As mentioned in the introduction slide, each phase of the cell cycle is regulated via protein kinase, the cyclin-dependent protein kinase, or CDK, and its subunit cyclin. Here, we see a graph depicting the variations of kinase activity for specific CDKs throughout the different phases of the cell cycle. We have here in G1, or gap phase 1, cyclin E-CDK2. It peaks near the end of G1 and start of the S phase, the synthesis phase. Active enzyme triggers the enzymes necessary for DNA synthesis to start to occur within the synthesis phase of the cell cycle. Then we have cyclin A, CDK2. It rises through, located here in the green, rises through the S phase, and then it starts to have a sudden decline as it goes through the G phase and into the M phase. It sharply goes down. And then lastly, we have in the red the cyclin B, CDK1, which dramatically peaks and then dramatically drops, all within the mitosis phase. The cyclin D will remain active throughout the presence of growth factors. However, this specific kinase is not represented in this diagram. The regulation of CDKs in cyclin is due to the phosphorylation and dephosphorylation of two proteins. The phosphorylation of thyrine or thyrine 160 by CDK activating kinase is required to allow CDK to bind to cyclin and become active. The dephosphorylation of tyrosine or tyrosine 15 of CDK removes the negative charge, blocking ATP, the energy, from the binding site. This activates the CDK to self-reinforce. The enzyme PTP is responsible for the dephosphorylation of the tyrosine 15, which is a substrate for CDK, and activated via phosphorylation. Both of these, the phosphorylation of tyrosine or thyrine 160 and the dephosphorylation of tyrosine 15 are taken into account with the activation of cyclin CDK manifolds occurs, allowing phosphorylation to occur again and downstream proteins target that are essential for the process of the cell cycle. By dividing this diagram that we see here in half, we have the top portion and the bottom portion. We will describe the step-by-step processes that are necessary for the regulatory mechanisms of CDKs involved in the cell cycle. So here we have that top portion of the diagram that we had just looked at, and step 1 shown here in the bottom left shows that the CDK is inactive when the cell enters the M phase or mitosis phase. Step 2, we are going up to the top left. The cyclins are synthesized, rolling into step 3 at the top center. This is where the cyclin CDK complex forms after the accumulation of cyclin occurs. The phosphorylation of tyrosine 15 blocks that ATP binding site, making the cyclin inactive. Step 4 over on the right top corner shows the thyrine 160 in the T loop and the dephosphorylation of tyrosine 15 of the CDK2, stabilizing the autoinhibitory T loop. This move away from the substrate binding cleft in kinase allows for the kinase to open, thus binding that protein substrates. This then finally activates that cyclin manifold. Step 5, located in the center here, depicts the activation of more CDK via phosphorylates, which then in turn activates an enzyme that dephosphorylates tyrosine 15. We then are rolling into the lower half of that initial diagram, and this is depicting steps 6 through 8. Remember that this entire thing from this slide to the last is all part of that initial diagram that loops and it shows one big cycle. In step 6, located here in the center, the active cyclin CDK complex triggers its inactivation via protein phosphorylation, and this is called a DBRP, or destructive box recognizing protein. We move over to step 7 on the right, showing that the DBRP and the ubiquitin ligase, which is another protein, attaches multiple ubiquitin molecules to that cyclin. And then and that's shown here with that little tau in the middle, bottom middle of this diagram. Then we shoot over to step 8 on the bottom left, and this shows that cyclin being degraded via proteasome and a proteolytic enzyme that makes the CDK inactive once again. Now that it is inactive once again, we can start this process all over again. As mentioned, regulations is needed to ensure that the cell cycle moves through the various phases properly. As mentioned earlier in this presentation, checkpoints are placed throughout the to prevent errors from occurring. The first checkpoint we've already discussed, but we're going to discuss a little bit further now, is located again in phase G1 before the cell enters the S or synthesis phase. And we can see that here on the diagram with this red bar on the bottom right. Here, the checkpoint ensures that the cell has grown large enough, made the proper proteins, and has adequate energy, thus creating a favorable environment for DNA replication to occur in that next synthesis or S phase. If conditions are not favorable for the cell to continue through the cycle, the cell will enter that G0 or resting phase to fix any problems before continuing on. That second checkpoint is located in the G2 or gap phase 2, just before the cell enters mitosis. This checkpoint looks to ensure that the DNA has replicated properly within that S phase. Lastly, there is another checkpoint found within the M portion of the cell cycle, or the mitosis, and this is an M checkpoint. Here, some refer to this checkpoint as the spindle assembly checkpoint. During the meiotic phase, metaphase is specifically where this checkpoint resides. So remember, this comes after the prophase portion of mitosis. This ensures that the chromosomes are properly aligned and attached to the spindle fibers, hence the name, the spindle fiber checkpoint. Understanding the phases of the cell cycle, the checkpoints, and other regulatory mechanisms is vital to understanding how cell functions is formed. However, it is also vital to understand that not all cells go through all checkpoints. Some fast track through phases while others take their time. Therefore, any disruption to these regular checkpoints or other mechanisms can cause damage, missing genetic material, and can lead to cancer or tumor formation. Knowing that cells need proper regulatory functions to ensure there is no damage or loss of genetic material, there are more than just checkpoints necessary for successful cell cycle completion. An important CDK substrate is PRB, or retinoblastoma protein. When the DNA is damaged, if you see here in the slide, PRB acts as a mechanism to stop cell division. This is done through unphosphorylated PRB binding to the transcription factor E2F. Movement from phase G1 to S is halted, and the combination of the PRB and E2F block is lifted once there is phosphorylation via the cyclin ECDR2. Looking at the image, we see ATM and ART on the top left. These are protein kinases that detect the break in DNA strand via signal protein from mRN at the breakage point. When the signal is received from ATM and ART, they phosphorylate something called P53, which acts as a transcription factor, stimulating the synthesis of another protein, P21. P21 is an inhibitor for cyclin ECDK2, and P21's presence ensures that PRB stays unphosphorylated and bound to the E2F, which continues to block the transcription factor activity and keep that cell cycle stopped in phase G1. By being in this state of arrest, the cell has the time necessary to fix any broken DNA prior to entering that phase S. This process and its mechanisms allow the daughter cells to form without containing any damaged genomes. The role of tumor suppressors and oncogenes. Tumor suppressors represent the opposite side of cell growth. They act to inhibit cell proliferation and tumor development. Inactive genes remove negative regulators of the cell proliferation. And oncogenes, these are mutated genes known as proto-oncogenes, and they can promote an uncontrolled growth and division due to unchecked activity of the encoded protein. This can cause cancer and can drive abnormal cell proliferation. Here we have an image where the to the right showed a side-by-side comparison of a normal versus a cancer cell. Considering what we've discussed thus far regarding the cell cycle and the checkpoints within, we see the intact checkpoints on the left. This example specifically shows MYC expression. MYC is a gene that encodes multifunctional nuclear phosphoproteins, which control various cell functions. So for this example, when there is an overexpression of MYC in normal cells, the balance is tipped in favor of the damage via stimulating the checkpoint mechanisms. This leads to cell cycle arresting and allowing for either repair or damage, like killing the cell. When looking at the cell, cancer cell on the right, showing that overexpression of MYC, the checkpoints are not intact and are actually in fact altered. Thus the DNA repair through homologous recombination occurs. This promotes the cell's survival and becomes resistant to DNA damage treatment in the future, such as chemotherapy and radiation. So if we're looking at this, we see an old-fashioned scale. Right here we have that center balance point. In the normal cell, it's tipped towards the DNA damage. It's recognizing the fact that this is damage and we need to either do something via our checkpoints and either fix it or kill off that cell so that it cannot be replicated and form more problems. When we're looking at the right-hand side of our cancer cell, we see that same balance. However, it is tipped towards DNA repair, which is the opposite of what needs to be happening, because all that is going to do is that's going to allow for a damaged and potential cancer cell to be replicated, thus continuing the growth of cancer cells within the body. It is important to understand cell cycle and all of its phases. The cyclins and the CDKs are vital to regulating the various phases of the cell cycle. Checkpoints ensure the cell is taking a time out, so to speak, to double check itself, making sure that, like we just saw on that one slide, if there are any issues, anything that is wrong, it can be fixed before it moves on. Therefore, taking away the fact that cell, either cancer cells or tumor cells or anything wrong with the DNA of that specific cell is not repopulated and replicated throughout the body. Dysregulation occurs when these checkpoints and the regulatory pathways aren't properly functioning, which can lead to more cancer and tumor. Ongoing research, specifically from Johns Hopkins Medicine, along others, is charting new molecular pathways to have a better understanding of the hallmarks of cancer cells. The research being done by scientists at Johns Hopkins and by other researchers is taking what we know about the cell cycle and looking at what goes wrong when certain enzymes are triggered. This may assist in the regulation that occurs to develop new therapies that can interrupt the cell cycle of cancer cells. The study being done specifically at Johns Hopkins University School of Medicine is looking to challenge the fundamental knowledge of the cell cycle and reevaluate the current ideas of how the cell cycle is regulated. Therefore, having a basic understanding really helps us have a better understanding of how cells not only grow, but also how they are policed, so to speak, so that we can further our understanding of not only the cell itself and its function, and understanding the fact that the division only happens during a certain portion of it, the mitosis portion, where the growth happens in that 90% of the interphase portion, but also how can we highlight and interject and intervene in these cells when there is this dysregulation that causes either cancer or tumor, so that we can maybe prevent the spread of these in a patient who is suffering from them. And here are my references. If you have any questions, please I look forward to hearing from them, and thank you very much for your time.